So hi everybody, my name is Hao. I'm a final year PhD student at Tufts. So today I'm gonna introduce you to RegDiffusion. It's a new class of a diffusion model to do very fast gene regulatory network inference from single cell data. So gene regulatory network, so gene network sometimes we call it, is a kind of directional network we use to describe how genes place with each other. And there are many ways to get to a GRN, but in this talk, we focus specifically on just inferring GRN simply based on gene expression data. This field has been actively researched in the past 30 years. We have seen a lot of methods being proposed, ranging from Bayesian network to decision trees, and also Bayesian networks. And in recent years, we are exploring this option on using deep learning methods to speed up this process. And on the other side, on the source of data, we also see a lot of advancements ranging from microarray to RNA-seq and to single-cell RNA-seq. So one of the problems we're facing today is like a lot of these network inference algorithms, they are very computationally extensive. And right now, today, we have exposed ourselves to a lot of data with a lot of genes and cells. These algorithms, they don't really scale that well. A few years ago, we had this method called DeepSum published. Essentially, this is an autoencoder-based structure equation model where they are trying to reconstruct the gene expression profile under the constraint of a parameterized gene-seeking matrix. People find that it's outperforming other methods based on the B-line benchmark, and it runs much faster than for example, like decision trees methods like Genie3. And one of the previous work that we have been doing is like we proposed a method called dropout augmentation inside this model called DESL, where we are trying to solve this, the dropout problem in single-cell data by introducing some extra amount of dropout noise. And we find that it's actually helping improving the model's robustness and stability. Before I move on, I would like to point your attention to this highlighted area where to model this agency matrix, we have to use this agency matrix on both sides of the autoencoder, both the encoder and decoder. So on the decoder side, we actually need to do this matrix inversion on the agency matrix. Unfortunately, this matrix inversion step, it's usually solved by Gaussian elimination, and it runs in cubic time. That says that if we have a single-cell gene expression data with N cells and M genes, the runtime will at least be n times m to the power of 3, right? So in this talk, we're going to show you how we actually managed to reduce the runtime all the way down to m squared. Again, we are using this diffusion model. I'm very excited to see so many diffusion model talks this afternoon. So basically, just a recap, a very quick recap. Diffusion model is widely used in those image generation apps, and it has two processes. In the forward process, you gradually added some Gaussian noise into the image, and in the reverse process, this is actually where the modeling process is happening. We're trying to predict the added noise from the noisy input. So as you can imagine, the reverse model, where we create the model, it takes two kinds of input: the noisy image and the diffusion time step t, which kind of tells the model how much noise should it expect. So the reverse model's objective is to predict the added noise, such that if we want to get the original image, we can simply subtracting the noise from the noisy input. In computer vision, people usually solve this with UNet, but we actually don't have to do this, as what you will see in this talk. So in RegDiffusion, we solve this problem by making this a linear additive assumption. Essentially, here, imagine we have 4 nodes, A, B, C, and D. And here we use X to denote the log of the probability of seeing A, B, C, and D. Okay? So the linear additive assumption just simply say that the XA will, the expected value of XA will simply be the weighted sum of X, B, C, and D. And because we normalize the data, we we want to make sure all these values are at the same scale. We assume the expected value of the weights to be 1 over, well, it should be minus 1. Yeah, 1 over the number of genes. If we write this equation in the matrix multiplication form, we have this very simple equation, x equals to xa. This is a matrix multiplication, and it's a very typical structure equation model equation. So then we ask this question: what will happen to this equation when we introduce some Gaussian noise? So we did some math, and we are very happy to discuss at the end. But here's the conclusion. So if we use x prime to denote the noisy input, then x prime a, well, when your expected value of the weight is small, then x prime a will be, in fact, be a close approximation of the original input x. So given this assumption, given this conclusion, we can see that if we want to calculate the added noise, we can simply subtracting this x prime a from x prime. Remember, in the diffusion model, I mean, those vision folks, they are using UNets, but the goal is to predict the added noise. This is actually exactly what we need. Here's an overview of the RegDiffusion workflow. As you can see, we start with raw counts. We did some pre-processing, and after some pre-processing, we get this x0, and we send this x0 to the forward process to noise the data, to noisify the data. And we send the noisified the data, xt, in the gray box, together with a diffusion time step t into the RegDiffusion model to generate the prediction of the added noise z prime. And the benefit of what I have been talking in the last 2 slides is that, as you can see right now, there's no need to do the matrix inversion. Here's an overview of the actual model architecture. It looks a little bit complicated, but it's in fact just a series of MLP. So we start with feature embeddings, MLPs, do some matrix modification, and then linear prediction. So no UNet, just MLP. OK, so that's the end of the model architecture. And to validate our idea, we tested on three data sets. We tested on the B-line single cell benchmarks, which itself has like 7 small, fairly small single cell data. But it also comes with 3 sets of ground truth networks. And we also tested on 2 microglia data sets at different sizes. The first one, which we call it Hummel microglia, is from the Best-Stevens lab. And it's these microglia cells, they are experimentally obtained. And the second one, I think, is from Bard. It's an Atlas experiment, and all these microglia cells, they are labeled by marker genes. And OK, on the right side is a evaluation metric, but basically the higher numbers means better performance. So RegDiffusion actually does great on the benchmark, but what really excited us is its runtime. And as you can see on all these B-line benchmarks, which I listed the size of the input data at the bottom. Most of them, they have roughly like 1,000 genes and 700 cells. Genie 3 took roughly like a little bit less than 3 hours. And DeepSam, the ensemble version, takes roughly like 5 minutes. But the RegDiffusion, it takes usually under 10 seconds on a GPU. And on the right side, you can see its performance on the Hummel microglia with 15,000 genes and 8,000 cells. The DeepSam ensemble version, it takes more than 40 hours GPU time. And we use a yellow bar to denote the compute time of RegDiffusion, but it's too short that you can't really see it. It's 3 minutes. So the source of speed gain is not only come from the elimination of the matrix inversion, but also it's because we actually use the fixed size bootstrap sampling. Basically, our single cell data, it comes with all kinds of sizes. Like different studies, they have different number of cells, right? It's a little bit unfair to send all this data and let the model see all the cells and still train them with the same number of steps. So so here we just simply we sample a fixed number of data, a fixed number of cells, and send them and let this thing go through the training network in each iteration. And then by observing the amount of changes in the adjacency matrix, we use that as a way to help us identify when the model actually converges. And what we find is that with the current setting, with the current default setting of RegDiffusion, regardless of the size of the data, no matter it's small benchmarking data or the real data, the bigger data we use, you always follow a very similar converge curve. So there's no need to see all the data in each iteration. Here, as I said, RegDiffusion actually does great on its on the actual prediction performance. There are two tables under different two different metrics. You don't have to read all these numbers, but in both of these two tables, RegDiffusion is at the bottom row, and we use colors to indicate the top performer or second performer. It's always, in most cases, it's always the best or the second best. Okay, now let's see some interesting biological results to inspect the actual networks that's getting inferred. Remember, in this study, we tested it on two microglia data, one from experimental obtained microglia and one from others with marker genes. And here what you're seeing is a two-hop neighborhood around ApoE, which is a very actively researched AD genes. And we use these colored ovals to mark out confirmed regulatory relationship relative to ApoE. Okay, so and you can see there. So there are, I mean, a lot of these links. I'm not saying like all of them, but a lot of them, we actually can find biological evidence. And another interesting finding is like if you compare the results, compare these inferred networks from ApoE from two studies, we can actually see the one using marker genes is in fact very coherent from these bottom left regions of this inferred network from the experimental obtained regions. So our theory is like, because microglia itself also has a lot of subtypes. And if you just simply use marker genes, it will only help you identify those major microglia subtype. So you basically throw away all these minorities in microglia types. Before I move on, I would like to point your attention to this green oval, the cathepsin group. Cathepsin is a kind of protease whose responsibility is to break down unnecessary proteins. In this case, in the microglia case, because microglia is one of those immune cells, right? So it's perfect to make sense to see the group of cathepsins like interacting with basically this gray area is microglia core genes. It makes perfect sense. And at the same time, we also find cathepsins in some of those deep stem benchmarks, like for example, the mouse embryonic stem cell. And in mouse embryonic stem cell, we find that cathepsin T, for example, is interacting directly with the gene BTG1 and 2. BTG1 and 2, after we did some research, they are actually anti-proliferation factors. So as you can imagine, these factors they help the embryonic stem cell to eliminate unnecessary like overproduced product when they actually grow. So our conclusion is that RegDiffusion, like when you apply it on the right data, it actually help us to capture different kind of very cell type specific regulatory strengths in different kind of cells. So here's our conclusion. RegDiffusion is very fast. It's very suitable for large scale data. And many of the links in the inferred networks, they are biological verifiable. And at the same time, they also comes with some kind of some limitations. So the we gain a lot of, we have a lot of speed gain in RegDiffusion, but it comes at the cost. I mean, there are no free lunch, right? It comes at the cost of the losing of the ability to do representation learning. And also, I mean, so far it's not capable to infer latent confounder node, just unobserved node. Also, at the same time, because there are a lot of feedback loops in biological system, we are not applying any DAG control at this moment. So if you check the network, you will see there are like a little bit too many like feedback loops. So we're actually exploring like possible options to do this kind of DAG control, but not entire DAG, maybe like few hop DAG control in a very in a better way. Yeah. And oh God, I forgot this. So, and we actually spent quite some time on developing a software interface for this. So this package is available on pip. It has a very simple like interface. You can simply load the B-line benchmark, start a trainer and do the training, and export the inferred networks, and even generate some visualizations you just saw using one line of code. And finally, this group picture. As I said, I'm a final year PhD. I'm looking for postdoc positions, hopefully in Boston area.
Questions?
Hey, good talk. In terms of like predict performance, have you thought about comparing with benchmarks on the basis of false positives and false negatives?
Yeah, FDR, and yeah, this is totally a valid metric. So far within the paper of that, we have not yet done that. But recently, we're in some recent work, we are starting to use this metric.
Okay,
yeah.
Cool.
Well, I actually have some actual comment on that, because like with one other thing I realized when we run all these kind of experiments is like our ground truth data, there are so many false positive and so many false negative. It's just hard to validate. Yeah.
Okay, thank you so much.